It is my honor to introduce today's distinguished speaker, Dr. Jeffrey Townsend from here. So in order to give this talk, Jeff drove all the way from here to Buffalo yesterday, right?
Yeah, that's right.
How long did it take to drive from here to Buffalo?
I didn't drive fast, so it took me something like 8.5 hours, but that's because I stopped and took walks on the way, which was nice.
Yeah. There's no direct flight from here, New Haven to Buffalo. So Jeff had to drive to New York City so it's
Much
okay.
better driving through New York City to JFK. Yeah. And
Okay.
it's very beautiful for money.
Yeah. Okay, so Dr. Jeffrey Townsend is an internationally recognized expert in statistical genomics, evolutionary biology, and cancer research. He's a Eliford professor of biostatistics and the professor of ecology and evolutionary biology at the Yale School of Public Health. So you're not in the medical school?
That's right. I'm in the School of Public Health.
Most
of my collaborations are with the medical school.
Okay. So Dr. Townsend is co-director of genetics, and epigenetics research program at the Yale Cancer Center, and recently completed his term as co-chair of the American Association for Cancer Research, AACR Cancer Evolution Working Group. Okay, so his interdisciplinary research applies cutting-edge methods from evolutionary biology, including theory on mutation selection, population genetics, molecular evolution, and phylogenetics, to challenges in basic science, public health, and medicine. He has made a major contribution to evolutionary biology, molecular development of mycology and ecology of human disease. In recent years, his work has focused on quantifying the fitness effect of cancer-associated mutation, revealing how mutational process and evolutionary forces shape tumor genesis, therapeutic response, and resistance. His integrative approach cooperates statistical modeling and large-scale data analysis to advance precision oncology and deepening understanding of evasion, treatment resistance, and cancer disparities. His research has been published in leading journals and has had a substantial influence on both public health and clinical practice. So today, Dr. Townsend will talk about computational genomics, statistical genomics, I guess, and the detection of and discuss the cancer driver genes and the pathway genes. Okay, so welcome, Dr. Townsend.
Thank you so much. Thank you, Eugene, for that gracious invitation and for the invitation to come here to the University of Buffalo to talk about the research that we've been doing. It's an honor whenever anyone thinks what you have to say is worth their attention to bring you to a place and witness. So thank you very much to all of you who are here and everyone on Zoom as well who's listening to this talk. So the title of my talk is Dismantling the coarse paradigm of cancer drivers and passengers. And my habit is usually to talk a little bit about the title before I go on to the rest of the talk. I'm titling it very intentionally as Dismantling. The point is the typical way that in our field of cancer genomics, people talk about the genes that lead to cancer, that have causal impact on cancer, is to categorize them as drivers, categorize them as passengers, say these aren't important, these are important. And then from that detection standpoint, move on to do experiments. And the argument I'm gonna make throughout this talk in like 7 different ways, you'll see all 7, I'll lay them out in the very beginning, is that that's not a nuanced enough way to look at it. And I don't think that that is a revolutionary thing to say. I think most people, you ask you, like, is that really driver passengers? That is as little as we should think about this. I think almost everyone would say, no, we should be more sophisticated about that. But I think the impetus for me to give a talk about it is, yeah, you need to know that that's true, but there's things you can do. There are ways you can act in the research that you're doing that act on the information that you should be acting on that is more nuanced than this driver-passenger division. So when you publish a paper and you say, oh, look, this particular site in this gene is hit 8 out of 20 times in a tumor, you're reifying this sort of like idea of, okay, well, it's just a matter of how much this thing is hit. It's a matter of whether it's a driver or a passenger. What you really should be doing is asking the question, how much does that elevate the growth and proliferation of the driver? And when I say that, I mean that like all of us need to be doing this when we're doing that. So that's my argument, is that we need to be a little more acknowledging of that fact and not just skip by it by saying, well, it's most easy for me just to call it a driver, call it a passenger, and move on, to actually use this quantitative information. So what is the quantitative information? That's what this talk is about. And I call it dismantling this coarse paradigm because, yeah, things are drivers and things are passengers, but sometimes they're drivers and other times they're passengers. And sometimes they're partially drivers and sometimes they're not really driving. And sometimes they're quantitatively driving a little bit and sometimes they're really frequent and doing a little bit. Sometimes they're really frequent and doing nothing. All of these are possible. So I'll give you examples of all this in this talk. I have a disclosure. It's not really relevant by talk, but I am a scientific advisor of Multi-Target Incorporated, which sells multi-target primers enabling multi-gene targeted reverse transcription. It's a great laboratory experimental reagent unrelated to the research I'm going to talk about. All right, so we can start a ways back. For a long time, we've been trying to understand the molecular basis of cancer. This is an early review in Scientific American by Weinberg, 1983, and they lay out. Weinberg lays out essentially the key things that we need to know in terms of causation of cancer. There are a couple others I could add, but the ones that Weinberg put down were point mutations happen that change particular genes in the genome due to mutagens. That mutation then leads to an oncogene. Oops. Well, normally I have a couple other points here. There should be gene expression also affects this. Also, there's copy number changes. There's a number of other things. The point of this slide, I don't know why the other ones aren't popping up, but the main thing that I want to emphasize is that I'm going to talk almost exclusively, maybe I'll have one slide at the end, I'm going to talk almost exclusively about point mutations. And I completely freely admit to you that that's because I'm the person late at night looking under where the lamp is brightest. Point mutations are where we understand everything the most. So that's where I can express the nuances of what I want to express in the most detail. I'm not trying to undersell all the other important things that happen in cancer and that are relevant. Copy number changes, which I'm working very hard on. Epigenetics, which I'm still thinking about working on, but there's a lot to be understood before I can apply the kind of expertise I have to that particular problem. So don't think that I'm ignoring those things. I do know they're all there. I do know they're all important. I'm just trying to show the greatest light where I can actually give you the greatest light. And so I'm going to lay out my thesis right in the beginning. What am I trying to say here? I gave you one introduction with the introductory slide, and here's another. In order to dismantle them, here are the things I'm going to tell you to do, basically. I'm going to tell you to avoid the statistical misstep that leads to drivers and passengers, which is reporting prevalence and p-value alone. So when you talk about, here's a bunch of set of tumors, and this is the frequency with which this gene is mutated, and here's a p-value for whether it's a driver, you're not done yet. And the reason is Biostats 101 says whenever you report a p-value, you should report an effect size. And the prevalence, or the frequency that a gene is mutated in tumors, is not an effect size. That's the patient population that's affected by the mutation. What you want to report with that p-value is some estimate of how strong a driver that thing is. And so if you're reporting a p-value and a prevalence and not an effect size of some sort, there's something else you should be doing. And it shouldn't be too hard to do that thing. And I'll tell you one way to do it, which is the way that I do it. I think it's the best way. There are a couple other approaches out there. Second, you should be computing context-specific strengths of selection on variants. So strength of selection is a phrase I'm going to use a lot. It's an evolutionary biology phrase. That's what I call the degree to which a mutation means that that cell gets selected among others to be represented in a greater proportion in the tumor, and that's why you see it when you take out a tumor and sequence it. So that strength of selection is a measure of the effect on a cancer cell, and it's a measure of how important a mutation is to the cancer cell, to its survival and proliferation. So you should be computing the context-specific strengths of selection on variants. I'll give illustrative examples in lung and prostate cancer, maybe melanoma, maybe something else, I'm not sure. I work on all kinds of cancers. These two are ones I've worked a lot on, especially. You should accurately and precisely prioritize cancer science by using these cancer effect sizers. So the cancer effect size, the strength of selection, this thing that I'm telling you that most people don't report performs better than SIFT and polyphen for telling you whether a mutation actually has functional importance. and it can improve AI and ML inference. And I'll provide data behind that. Lastly, you have thinking about what drivers and passengers are, we need to account for dynamic changes in selection that occur across the process of oncogenesis, that occur as other mutations arise, that occur as the microenvironment changes in various different ways, and that occur in response to therapy. So it's a really dynamic thing. It's not like you can categorize it, driver, passenger. It depends on the patient, what the patient is doing, what therapies they're receiving. All of these things make differences to what it is to be a driver or a passenger. So that binary classification is really a weak way to think about it, and whenever you can, think about it in a more nuanced way. Okay, so that's my argument, and now I'm gonna give you the data to try to back it up a little bit and persuade you. First, avoiding this misstep of leading to just drivers and passengers, prevalence and p-value. So in 2013, in a really groundbreaking paper, Lawrence et al. Published the first analysis that actually did a good job of calculating the p-values in the first place. Like the very first papers just looked at tumors and were like, this is frequent. Boom, I'm done. They said, we really should have a p-value for that. We figured out p-values. Lawrence had 2 papers in 2013 and 2019 that did a great job calculating p-values. They didn't report prevalence and didn't report cancer effect size, and I'll get to that in just a moment. But the thing that I would just emphasize is that different cancer types have very different numbers of mutations, both within cancer type and between cancer type. This is a log scale, so we're talking about very much bigger numbers up here than down here. And so those differences in tumor burden mean that you sometimes, especially in these cancers with very large numbers of mutations, are getting a lot of mutations which are what people typically call passengers. That is not really affecting the course of cancer evolution. And you can see mutational processes underlying this. So these are distributions of how many of these mutations are C to T, C to A, C to G, T to C, T to A, or T to G in each of these cancers. And you can see it's very different for different cancers. Way up here in the high mutation range is melanoma, which is almost all C to T changes. That's because UV light causes C to T changes in your skin. Almost all of melanoma is caused by UV light. You'll see that again later in this talk. Okay, so we've got this idea. How did Lawrence et al. Calculate their p-value? Well, they said, well, we've got to account for the fact that different sites have different genes and different sites have different mutation rates. And what they did is they looked at how mutation rate varies across the genome, and they showed that it correlates with DNA replication time and expression level, also chromatin marks it also correlates with. And here you can see sort of this, the co-correlation between the mutations per megabase in red, between low to high expression, and between late and early replication time. So there's a high co-correlation indicating that these cofactors or covariates are pretty highly determinative of the mutation rate, not necessarily in a causal way, although there is a causal reason for expression, which is transcription-enabled repair. Okay, so, and one of the things you see is that some of the genes that people early on found and were like, what, that's not a cancer gene, why is that there? Show up as being present on these very, very high mutation areas. So olfactory receptors, for instance, which no one really believes are causing much cancer, tend to be mutated a lot in tumors just because they're mutated a lot. Because in most tissues, olfactory receptors aren't expressed. There's no matter if it gets mutated, and they get mutated. CSMD3 is another one, very high mutation rate. And this is just to show that these correlations between expression level and mutations per megabase, between replication time and mutations per megabase, and I'll skip this for the interest of time. Okay, so now we can calculate p-values by saying, do we see more mutations than we expect given the underlying mutation rate? Pretty straightforward idea, and this is the kind of result that Lawrence et al. Reported. This is in 2014, this particular paper. And what you can see is that for these different cancers, we now have much bigger numbers than these in many studies, but this is still representative. For each cancer type are reported the p-value on the x-axis here, and the prevalence by the size and color of the lollipop. And what you can see is that some prevalent things have very high p-value. Generally speaking, you have more power when you have higher p-values, but then you have some things like the CBPA, very low p-value, but very high prevalence. And that is a little hint there that there's some other story beyond what this is showing you that we ought to be thinking about. And in general, this is the kind of way, since this time, most data is reported, p-values and prevalence. And I'm gonna argue that this is great. There's nothing wrong with what they've done here, except they aren't reporting the most important thing, in my view, which is how important is it to driving cancer? Because a p-value is a measure of belief. And again, the prevalence is the patient population. Neither of those is how important it is for cancer. So what do we do? Those are the two things everyone reports. I'll just point out that even in 1976, sort of conceptually, some of this was understood. Noel's very original paper in Science in 1976, introducing the idea that cancer was a consequence of somatic mutation, included the idea that along a cancer lineage, certain cells die off, and certain cells manage to replicate more, and the ones that replicate more end up taking over and lead to malignancy. That's an evolutionary process, and that's called natural selection, somatic selection in this particular case. And measuring that selection process is one thing we have to do, and measuring the mutation process, where the mutations occur, is another thing we have to do. And what we have to do is disentangle those two things to understand this better. Because if something mutates a lot, that doesn't mean it's important for the growth and replication of something. If something is present in a tumor at high frequency, then maybe it is. But let's figure that out. Okay, so I'm going to skip through these just to make, for time, and then just, sorry, here we go. Why is this backwards? Yeah. So I've made the argument that there's a statistical misstep, which is skipping effect sizes. I hope that you think through that anytime you're looking at papers, evaluating papers, and writing your own papers. So what are we going to do? Let's solve this problem. We'll compute context-specific strengths of selection on variance. And the way we're going to do this, I'm first just going to motivate a little bit by, if you think of the mutation rate for something on this x-axis, and you'll have to just trust me that using sort of Lawrence et al. Style methods, we're able to estimate the underlying mutation rate, and the prevalence, which is easily reportable from any tumor sequencing project of a given mutation. What do you expect the picture to look like if you look at all the different genes in terms of their mutation rate and their prevalence? So make a little picture in your mind of what you think that's going to look like. It appears none of my animations are going through. Oh, there it is. That's what it actually looks like, okay? Why does it look so different? Why isn't the mutation rate causing the prevalence? Well, that's the action of natural selection, because many of the mutations that occur, here's an odorant receptor, here's another odorant receptor, they occur at some relatively high frequency in some genes. Most of them are happening at relatively lower frequencies, but what you see at very high frequencies, these major drivers, are there because of selection, not because of underlying mutation rates. But it's really important when you start looking down at this corner here to differentiate between the things that are there because of high mutation rate and the things that are there because of high selection. And that's what I'm going to spend the rest of my time here doing in various contexts. So how do we quantify this cancer effect size or strength of natural selection? To do it, that prevalence, how frequently something is seen in a tumor, has to be deconvolved into the baseline mutation rate and the degree of selection for the mutation in the cancer lineage. So the basic principle here is mutation creates variation within a population. Unfavorable mutations are selected against. Reproduction and mutation occur. Favorable mutations are more likely to survive. And the consequence is, down at the bottom, the frequency with which you see things is dependent jointly on how much mutations are creating that variation and how the selection operates on that mutation. So both of them contribute. And so we need to take them apart and reproduce, yeah. So mutation rate at each site. So if we can calculate one, we can calculate the other, because they're sort of working together to lead to the prevalence. And we can calculate the mutation rate the same way Lawrence et al. Did, essentially, with a little bit more elaboration so we can be even more precise. Mutation rate at each site in the genome can be calculated by convolving the gene-based rates from silent sites and the covariates, and those covariates I talked about from Lawrence et al. With the trinucleotide context. I think my battery is low on this because it's not advancing very well. Is there another way I can advance? Yeah. I don't see a keyboard. Where's it? There. Okay. Thank you. All right, great. So this is just a unidimensional plot, even though it's jittered, so you can see it a little bit better. The point is, if you look across the genome, what are the different mutation rates of different genes? This is analogous to the Lawrence et al. Paper I showed you, but using our mutation rate method, which is a little bit more advanced. And the point is just to say that different genes have different mutation rates, and it varies over 2 orders of magnitude. And that's really important because if you think about what that means, if you've got mutation order varying over 2 orders of magnitude, there can be 2 orders of magnitude difference in prevalence that just comes from mutation and has nothing to do with whether it's causing cancer. So this is really important. And that it's even compounded by the fact that in addition to that gene by gene mutation rate difference, you have enormous differences by trinucleotide. So each site has different mutation rates within that. And what that means is that you have another 2 orders of magnitude of variation by mutation rate in terms of site to site, which means that when you actually look at predictions of whether certain sites should be hit, and this is just like different amino acid changes occurring over, I don't know, like 15 or 20 base pairs of 3 driver genes that we know about, we get vastly different mutation rates on a gene by gene basis, but also on a site by site basis within each gene. And so we need to use that to calculate the underlying strength of selection and not just say, oh, this is a hot spot, it's got a ton of hits, therefore it's a very important spot. That's just not the right inference to make. Okay, so we can do that for these different genes. Here it's KRAS, EGFR, and catenin B. And when we do, we see the following thing, which is that we don't see the pattern that's described by mutation. We see a different pattern in tumors. And that's because, at least at these sites, because these are actually selection-driven sites, some sites are way more common than they're expected to be in this pattern, and some are less. And that's because of the action of natural selection. So this sort of illustrates the sieve of selection, at least the way that term works. We're selecting the variants that, these variants all happen in the genome, but these are the ones we see spread across the tumor when we sequence a tumor. And that's the selective sieve of natural selection. So all we need to do to really figure out what the strength of selection is to sort of divide the strength of how frequently we see it in this box by the prediction in that same box under mutation alone. And it's a little more complicated than just division, but I'm not going to go into the statistical complications. Depending on the site, it may mean that certain sites are really, really strong drivers and certain are weaker drivers. This is L858R in EGFR, major lung cancer driver in non-smokers. This is KRAS G12, major driver in smokers. This is catenin B1. No one of these sites is actually all that strong a driver, but there's a bunch of them. And so catenin B1 ends up being relatively important as well. Okay, now, once you have that site-specific mutation rate by dividing those one by the other, you can do all kinds of fun things. And so there's a big jump here, so see if you can stay with me, I hope you can. At each site, we can calculate that genomic mutation and cancer causation and specifically attribute it to different causes of cancer. So if you can deconvolve the mutational signatures in different cancers, and then you can see, oh, smoking causes most of the mutation in cancer, that's the brown one here, along with some by defective homologous recombination, APOBEC, and some aging processes. So this is the underlying causation of cancer here, just represented by the width of these. But then you can look at every site and characterize, oh, which cause is most likely to have caused the L858R, the G12C, et cetera. And you can see G12C very strongly indicated by smoking. That's why we see a ton of that mutation in smokers, hardly any of it in others. L858R, hardly ever caused by smoking, mostly caused by other processes. That's why I see it in non-smokers. And this then gives us a picture for what the specific causation of every variant is based on its underlying mutational signature. We can do it for other cancers. This is bladder cancer and cervical cancer, where it appears that APOBEC is causing a lot of these. APOBEC is an internal process I'm not going to talk about right now, but I'll talk about anyone who's interested in it. In these cancers, but specifically PI3 kinase mutations in both cancers are strongly driven by this APOBEC process. So as I said, we can calculate this on this entire genome, since we can calculate underlying rates on every genome, and we see the prevalence in the sequencing of how frequent different variants are. We can calculate the exact effect sizes for every variant that we see in genomic data sets. And this is just to, you don't have to read any of these, but just to emphasize that you can do it across many different cancer types. And this quantification helps us to prioritize what we should be targeting in terms of drivers, what we should be researching, et cetera. Some of the big drivers, of course, are not total surprises. KRAS up here, G12 mutations, very, very strong drivers. Up here, BRAF on the right. BRAF in thyroid cancer and melanoma, very strong driver. Anyway, you could look at this for quite some time and be entertained by seeing all the major drivers that people have seen, plus some drivers that people don't typically see. I just want to emphasize that the distribution of selection kind of looks something like this. These are just some examples of those drivers I was showing in the last. There's actually a bunch of them out here. This is a long tail here, but most of the mutations in the genome are not really driving selection. So that's the traditional passenger distribution. But there's a long tail of ones that are causing it. And that's true for all of the different cancer types. Okay, not only can you do this sort of on a cancer type level or on a specific site level, but we can do this for individual patients. So each one of these double bars represents one patient. This is what the mutations that are caused in their genome, and this is what's causing their cancer. And in some cases, those can be identical. In this case, there's an individual who has skin cancer, and nearly all of their mutations in the genome were caused by UV light, and nearly all of their cancer was caused by UV light. And that's because only a few mutations are actually causing the cancer. So we have to filter through those few mutations to see what's actually causing the particular cancer. On the other hand, in some patients, here's an example that's the exact opposite extreme, where most of the mutations were caused by aging and not UV light, and yet more of the cancer was caused by the UV light than by aging. And this is skin cancer is the one where they're most consonant because UV light causes all the mutations pretty much, so very rarely do you see differences. But if you go to colorectal cancer, HPV negative head and neck cancer, and then thyroid cancer, you get bigger and bigger differences between what's actually causing the cancer and what you're actually exposed to. And so it's really important to know that because in terms of prevention, we want to prevent the things that are causing cancer, not the things that are causing mutations that don't do anything to us. All right, and the mutation from a source is not necessarily proportional to its cancer effect. You can, this is not the best way to show it. I'll show you in a slightly better way in the next slide, but this nicely organizes things by how much is caused by aging, how much is caused by these other exogenous things that we could maybe prevent or treat, and how much is caused by factors that we don't yet understand in black. And what you see down at the bottom are things like glioma, largely age-caused, thyroid cancer, largely age and apoptotic-caused, glioblastoma, also largely age-caused, way up here to melanoma, which basically if you stay out of the sun, you're not going to get melanoma for the most part, with a few exceptions. And smoking for lung cancer. If you don't smoke, you're much, much less likely to get lung cancer. So you can really see that just from the genomic data. We don't have to do an epidemiological study, and it's probably more accurate than measuring pack years of cigarette smoke, frankly, because that's not a very accurate way to look at mutation. Okay, so this just shows you the differences in an animation between what you predict based on just looking at the genome and how many mutations are there, and what you see when you actually look at the causal effects. And so just to emphasize that those are different and they matter, especially on an individual patient case. Okay, so I've given you some illustrative examples of the context-specific strengths of selection on variants. And this is just one more that's on a transition to quantifying these in a more quantitative way. The number of low frequency mutations that exhibit high effect size in lung adenocarcinoma. So what I want to do here is just point out the two major drivers, KRAS and P53. If you look at the fixation rate, which means essentially the same thing as the prevalence in the population, it's proportional. KRAS and P53 are clearly the strong drivers, and everything else is sort of all over the place. If you look at the underlying mutation rates, though, those are kind of different. So for instance, the mutation rate for KRAS is a lot lower than P53. That's not surprising. There's so many sites in P53 you can hit. These also vary across different, the other mutations as well. And then if you sort them instead by the scaled selection coefficient or how much they are important for driving the cancer, you see how much more important KRAS is, for instance, than P53, and how much more P53 is important P53 is than almost everything else. But then as you look down this distribution, look at the size of those circles. It varies all over the place. What that tells you is the prevalence is not giving you a good ranking for how much those mutations cause cancer. You really need to do this analysis. And you get some small ones like this one. Here's Catenin B1, which I showed you earlier. Not that frequent in lung cancer, but when it's there, it's causing a lot of the oncogenic effect. And so a drug, for instance, for the rare patient population that has Catenin B would be particularly effective. All right. I just want to emphasize how much, there's a lot that we know. Some people are like, oh, we're done sequencing. We've learned all we can learn from tumor sequencing. I totally disagree with this point. This is a paper in 2018 arguing this very point. Armini et al. Took 1,000 prostate tumors instead of 500. And when they looked at the sequencing from that. They went from having, I think, 13 drivers, which are approximately the top one, 13 here, to over 100 that they could show statistically were driving things. I went on to analyze that same data set, so I'm not actually gathering any more data. I'm just using the cancer effect size approach to look at different variants and whether they're statistically significant or not. And you can show that there's a ton of variants that are not statistically significant. So if they have a star, they're statistically significant and would have showed up in Armenia's plot. If they don't, then they wouldn't show up as statistically significant. But our estimate is still that they're strong drivers. And these include things like splice site mutations you don't see very frequently, but are causing a lot of, the estimate is that this NOS2 mutant, which after 1,000 prostate tumors, we still haven't seen at a level to call it significant. The estimate is, it's not statistically significant yet, but the estimate is it's among the top 10 drivers of prostate cancer in terms of the strength of driving it in an individual patient. So there's a lot for us to learn as we keep sequencing tumors. It's not something that's saturating. Also, I just want to point out, CUL3 is something that Armenia et al. Detected in their going to 1,000, but look, here it is, very high in terms of strong driver of cancer, and that's not surprising. Other research in molecular biology has shown it is as well. So we're finding strong drivers as we go on, and that one is statistically significant. And I just want to hammer this point home, I'm going to talk a little, if you are a structural biologist who sort of says, well, show me some real structural biology that will persuade me. Here are the driver mutations of the SPOP gene, which is one of the major drivers in prostate cancer, ordered by their cancer effect. And there's 2 points I want to make is, different drivers in a given gene have very different effects. So you see a big variance here. This F133V is a very strong driver of prostate cancer. F102L is significant, but it's just a much weaker driver. And then I've colored this by a gradient here and then plotted the color of the strongest variant at any given site with its original amino acid there. And this is the BRD3 ligand that is bound by SPOP. And you can see by how closely interacting these amino acids are with that ligand, how strong a driver it is of cancer. Because we know exactly the mechanism SPOP works is by essentially obtruding or limiting the ability to bind this ligand. And so it plays out just beautifully with what we're getting purely from sequence data here, that those drivers, those mutations that occur very close to that BRD ligand are the ones that are actually driving it the most, and the ones that are a little farther away drive it less, and the ones that are really farther away aren't drivers at all. You can see, this is a little harder to see. Sometimes these structure plots are very hard to get an angle on, but if you had 3D and I could spend all the time, I could show you that all of these are ones that interact with the ligand for the androgen receptor as well. So again, the closest interactors here are the strongest drivers. The ones that are farther away are the weaker drivers. So we can accurately and precisely prioritize cancer science by this. What I want to just say here is that they're a key metric and they play a lot of different roles. So they should prioritize your basic research. If you're interested in doing basic research that serves cancer, if you're working on pathways that have very high effect, you're going to probably have more downstream effect on actually helping out with the translational science. In terms of precision medicine, this can be used today in a tumor board where you have 2 different treatments that are used off-label because they're for some other cancer type, and you don't know which one to use. Maybe they're equal in every other way. You want to use the one that targets the variant that actually has higher effect because it's more likely to actually lead to a better outcome. Clinical trials, we can't do every clinical trial we'd like to. We should do clinical trials on treatments that hit things of high effect, not on things that hit things of low effect. And pharmaceutical development, of course, we want to develop drugs that target these things that have high effect. And for doing so, this measure performs better than SIFT and polyphen. These impact scores are very standard in risk prediction. They're used by all kinds of bioinformaticians. Cancer effect is not. I don't know why not. It's just not. But impact scores, they're frequently derived from observations of evolutionary conservation. And in order to do what we do, we deconvolve these baseline mutation rates. I told you that already. But these scaled selection coefficients, what we did to just test this in a very straightforward way is look at whether the scaled selection coefficient was a better predictor for whether something is a ClinVar variant. These are clinically validated variants. And the scaled selection is a much higher for those that are selected, for ClinVar variants than non-ClinVar variants. So if we just look at mutations that are recurrent within the cancer type, you can see much better you know, the distribution is almost bimodal between those things that are ClinVar variants and those things that are the scaled selection coefficient is much higher for the ClinVar variants than for other SNVs that are recurrent. And even if you look at ones that are just hit once within a cancer type, so we don't have much statistical significance here. We're just looking at ones that are hit once within a cancer type, but where we know stuff about it clinically because they're ClinVar SNVs or not, the ones with higher selection coefficients are way more likely to be ClinVar SNVs than other SNVs. So we should be using cancer effect size. And a more statistical way to do that, we can just look at the predictive power of it. Cancer effect sizes are more predictive, labeled on ClinVar status than impact scores. So if you look at whether the predictions are true or false based on CES or these impact scores, you're going to do way better with the prediction from cancer effect than from the other ones. Our sensitivity is 0.25, specificity is 0.99, accuracy is 0.996. And then one other way, maybe the most sophisticated way to do this is to do a multiple regression based on a bunch of different predictors such as mean cancer effect or top cancer effect. And then you also include CIF score, polyphen, or variant prevalence. And if you look at these numbers here, the important thing is how big is this number, not whether it's negative or positive. How big is this as a predictor for whether or not something is a ClinVar variant or not. And the mean cancer effect is way higher than anything else. CIF score, polyphen, mean prevalence. And this is a multiple regression. So if CIF score had something to explain about it that cancer effect didn't, that would appear in here, but it doesn't really have much to explain that cancer effect doesn't. And I've skipped through it. Also, there's this stuff, which I already talked about. You can extend this to modern AI tools, which are being used for predicting variants now. This was done by Nick Fisk, a finishing graduate student in my lab, who's now a professor at University of Rhode Island. And so I don't want to spend a whole time going through the approach here. It is all written down on the slide, but basically what we did is just test SIFT and polyphen2 and CES for use in a binary classification task of clinical features. We used 21 different data sets, 76 different tasks using 3 different AI approaches. So very just like throw everything at it and see whether we do better with CES than other approaches. And the bottom line was that cancer effect size in every single permutation of this, and we did not select them, we just chose a whole bunch and then ran it, it outperforms these functional impact scores and performs as well as all 3 in combination in every single instance, or performs as well as all 3. So this is, it's doing it, it achieves as well as it possibly could. This is, it achieves better than anything else. It non-max ties, that means it does as well. And you can see across all these tasks, many tasks it does the best it possibly could, and some, but it always at least matches the other ones and usually does better. I'm going to skip this just for the sake of time because I'm going to run out of time. But the point that there's a whole bunch of slides showing we can do better throwing in CES than other factors into any sort of regression by AI. Any type of AI tool, if you throw in a cancer effect instead of the prevalences of the mutations, you're going to do way better. All right, we also need to account for dynamic changes in selection across oncogenesis. So mutation rate actually increases with oncogenesis. That's because as you mess up the cells get messed up more. It's sort of a catastrophe in the weighting. You can look at the density of different mutation rates. This is looking at lower risk tumors compared to higher risk tumors in prostate cancer. And then higher risk tumors, and then here we're looking at metastases versus primary tumors. And you can see the distribution shifts to the right, meaning we have higher mutation rates later in cancer evolution across oncogenesis. So we need to account for that. And we can account for that, and we can actually calculate selection looking across those different stages. And what we see is that selection on new mutations changes dynamically across oncogenesis. This is an example from prostate cancer. You can see CUL3 very important in early stages, but not so important in mutagenesis. And what I mean by important is these mutations, if they happen early in mutagenesis, have a big cancer effect. If they happen toward metastasis, they do not have much of an effect. So we no longer have selection for CUL3 mutations later in evolution. SPOP, same way, it's an early driver. Most of these are early drivers. A lot of the mutations happen in early, but we have a few that are later. For instance, P53 stays important later. Ketinin B1 stays important later. PI3K, it's not quite significant, but it seems to become even more important later. And androgen resistance, not surprisingly, is only important later because it's actually caused by selection in therapeutic resistance. Okay, so there's dynamic changes that happen across stages, and my argument would be, and I have a lot of data that I can't show you today, but that this is an interaction with the other mutations that arise. So for instance, you have one mutation, it occludes other mutations from being selected, et cetera, and that causes some of these stage-specific differences, but also means that, that things have an epistatic interaction. A lot of the time, what you see in papers is people talk about mutual exclusivity and co-occurrence. These are empirical measures, and you can show them in big plots, so people like them a lot. But they have not, but generally speaking, they're not, I mean, there's better ways to do it. These are not acknowledging the possibility of a co-mutation, or in other words, a common underlying mutational bias. I told you there's all those mutational processes that happen. If a mutational process causes one mutation and another mutation, very likely, then you'll see correlations in the tumors, because ones that have a common mutational process will have a common mutation that occurs. And so you'll see this correlation between having one mutation and another. It have nothing to do with them actually interacting. It can have nothing to do with them actually interacting. So mutual exclusivity, co-occurrence are not the right measure for understanding whether things are interacting. To do that, you need to back out the underlying mutation rate, so you're not biased by co-mutation within particular tumors. Remember, mutation is different from variant in tumor. Mutation means happening in a cell. Variant in a tumor means it's spread already throughout a tumor. Okay, so what do we do instead of that? What you want to do is calculate epistasis, and the same software that is, you can download from my website calculates the effect size. It also calculates this epistatic effect. So pairwise selective epistasis underlies the changes in selection across oncogenesis. This is looking at that prostate cancer data set again. Here's the epistatic change in selection, meaning how much is the selection changed when the other gene is mutated. So this is SPOP gene pairs, and the question is like, what change in selection for the mutated what changes there in the selection for mutated SPOP after this gene below is mutated? So when P53 is mutated, SPOP is less selected. AR is mutated, SPOP is less selected, et cetera, et cetera. So SPOP must be an early gene, because every other gene, if you mutate it, it lowers the selection for SPOP to occur. SPOP has a strong selection to occur to start, but as evolution of that tumor goes on, it gets lowered by all these other mutations that are happening, with a few exceptions of RhoA and Ketinin B1, where it actually has some interaction where SPOP being mutated, SPOP is more likely to be mutated after them. And you can go through different genes and see these same patterns. And this explains like why we see the patterns that we do of mutual exclusivity and co-occurrence, but allows you to estimate the effect size of those changes and therefore properly figure out whether they're interacting or just caused by mutations. And you can build together oncogenetic trajectories. And I'm going to show you better examples of this later. This is just drawn out using a lot of data and figuring out in the head. This is sort of a conceptual diagram, but SPOP playing an early role. You can see AR and PI3 kinase playing a later role, and other genes playing other roles in low risk, high risk, and metastatic prostate cancer. And we can do this quantitatively. So these are all the different genotypes in lung cancer now, work by Jorge Alfaro Murillo and my group. Normal tumors, P53 mutated, KRAS muted, STK1 muted, LRP1B mutated, and all the other permutations. And in every single one of these, we get a different underlying selection coefficient for the different variants. So it matters what was mutated before, how strong the selection is afterwards. And then you can depict this for a given tumor type in this kind of diagram. These are all the normal tumors. I don't mean that they're normal tissue. What I mean is they're this should be really a different terminology, but that just means they're tumors that don't have any of the 4 mutations we're talking about here: KRAS, P53, STK11, and LRP1B. So we have quite a few of those that have none of them, but then we can see the ones that have STK11, et cetera, et cetera. And these arrows indicate in this case, they indicate the strength of selection for these different mutations. And what you can see is that it depends on where you are in this sort of landscape of evolution, what the selection coefficient is. So KRAS is reasonably strongly selected here, not so strongly selected after P53. P53 is But here, where we have p53 and LRP1B, it's actually especially strongly selected. So this gives you hints as to like precision medicine, like when is a KRAS inhibitor going to be most valuable? It's going to be most valuable when the selection strength is strong. And just to break that apart a little bit more for you, you can break that down into underlying mutation rates and selection here. So this is the really important data. This is when they're actually selected as opposed to what the frequency of their mutation changes, which is the product of the mutation rate and selection. And you can see there are these very strongly selected routes that are what we want to stop in designing therapies that would stop the evolution of cancer. And again, I'm running low on time, so I'm going to skip past this. I'll talk about how selection changes as the microenvironment changes. So for instance, mutation rates of genes vary between ever smokers and never smokers. So it's not just genetic changes, but environmental changes that make a difference. Here's the mutation rate in never smokers. Here's the mutation rate in ever smokers. Obviously, mutation rate in ever smokers is going to be higher than never smokers, but here's the visual evidence of that. But it varies by gene. So ALK has a much higher mutation rate in smokers compared to, say, ARID1A, ketinin 1B, et cetera. So the underlying mutation rate is different. And I'm not just saying that how frequently it is in tumors. That's like the cellular mutation rate is different because you're smoking. It's a very different tissue when there's that chronic exposure to smoke. Not only is the mutation rate different, but the selection is different. Like the chance that the mutated cell actually spreads throughout the tumor, it depends on whether you're a smoker or not because it changes the environment of the smoker. So for instance, here's what you might, the pattern you probably most expect is like in smokers, it's actually this KRAS G12 mutation I told you before has a higher mutation rate. It does have a higher mutation rate, but it also has a stronger selection. So you're more likely to get the mutation, and then when the mutation appears, it's more likely to spread within the tumor. And there are a number of other ones, KEAP1, ATM, ARID1A, et cetera, that are all in that territory. Here's the sort of astonishing thing, though. There are things that are more likely to spread in a normal human tumor tissue than in the smoker tissue. The mutation rate is lower, but when they happen, they're more likely to spread in normal tissue. And in particular, here's EGFR. Everyone talks about how EGFR-driven cancer is what you see in a non-smoker, and here's the selection. It's much stronger selected in someone who's not smoking than in someone who is smoking. So there's something about the smoking process that changes the physiology of the lung. It means that it's really hard to drive that EGFR. We have a paper that's going to come out soon. We have some hypotheses about why that is. I'm not going to go into it today, but we did transcriptome analysis, and you can see some differences in EMT that are relevant to that. So dramatically different results. It matters whether someone's a smoker or not. And this is the coolest result. When you look at both the KRAS clinical trials and the EGFR clinical trials, you see exactly this pattern. So for instance, when you use an EGFR inhibitor in a smoker, those are rare cases, but it doesn't do them much good because it's not even selected. It's not really causing cancer in them, even though they have that mutation. And when you use a KRAS inhibitor in a smoker, it does more than in a non-smoker. So the patterns that we're seeing here are reflected in human clinical trials. Again, I'm running out of time, so I'm going to skip over this. Just to say, you can look at G by E variation, G by G variation, G by E. It all exists. There's all kinds of variation in how selection happens, and it can get very complicated. So another really cool thing, if you look at how mutations, what the selection is as people get older, here's age on the x-axis, discretely or in sort of a continuous functional fit. And what you can see is these NRAS genes, they're stronger selected as people age. So the tissue of the aging person in some way means that when that mutation occurs, it's more likely to spread throughout the tumor than when you're younger. So it's not just that you're accumulating mutations that causes cancer. This has been a long-standing thing people have thrown about hypotheses about. This is like real data justifying the fact it's not just that. Your aging tissues actually are not as able to compete with the cancer cell lineages, at least when it's NRAS driven, because here's a surprising result. When it's BRAF driven, you're actually less likely to have the selection is less likely to be driven by the BRAF mutation. So you still have problems with getting more cancer when you're aged. That's largely driven by the NRAS mutations that are occurring, but actually you're less likely to get cancer from BRAF, even though you're more likely to get a BRAF mutation as you're older, more likely to accumulate mutations. Okay, last point I want to make is that in response to therapy, you can see the same thing. So selection on changes they're dynamically in response to therapeutic intervention. This is a phylogenetic tree of many metastases and a primary tumor. This is an individual who had their primary tumor extracted, was given permatrexib, cisplatin, and then erlotinib. And we can see on the link, based on the sequencing of all of these, that an EGFR T790M mutation appeared somewhere on this lineage. Because of this is a timed tree, so we actually use evolutionary methods to figure out the actual times based on when they had their surgeries, the progression of disease, et cetera. And there's some error on it, but generally speaking, this is the best estimate of the timing. Because this is a gene we know mutates in response to the treatment of erlotinib, we presume that although it's anywhere on this branch, it probably happened right up here, which means it happened extremely quickly. And the interesting thing about that is that it's not surprising it happened extremely quickly because this person was treated with cisplatin. And if you remember those plots I showed you earlier, I don't have time to go through it, but if I remember those plots I showed you earlier, this mutation is particularly likely to be caused by cisplatin. So basically this treatment regimen of giving cisplatin and then erlotinib predisposes the patient to get the resistance mutations before you give them the erlotinib. So do not do treatment in an order of cisplatin and then erlotinib. Erlotinib is an old drug, it's not being used right now, so this is not so clinically relevant, but it's still used sometimes, and if they've already had cisplatin, it's probably not gonna be terribly effective. That's an external process. I'll just mention one internal process as well. This is a cadherin B1 mutation, and what's interesting about this is that this Avastin that was given, you can see again that EGFR T790 in response to erlotinib, but it took much longer for that to happen because this person wasn't good with cisplatin. But later on, they got a cadherin B1 mutation, which causes invasive, I always forget this word at this time in this talk. The word for when lots of capillaries form. Angiogenesis, thank you. Angiogenesis, thank you. It causes angiogenesis. And what's interesting is all of these tissues, all these metastases have that mutation, but this one doesn't. Can anyone tell me why that pattern might arise? If you learn about the spleen, what's the one thing you learn about the spleen biology? Not many people learn about spleen biology, so I forgive you if you don't know. Spleen is incredibly blood infused. There is enormous amounts of blood around the spleen all the time. So you don't need to have angiogenesis for the cells to survive in the spleen. So I just love how you can see the biology on the phylogeny of this particular pattern. Again, I'm running out of time. I just want to say you can also do this on treatments, and that EGFR T790M mutation is stronger selected than even the original, EGFR L858R mutation. Why is that relevant? That's relevant because that means we're putting an enormous amount of pressure on that cancer to survive when we give them erlotinib. Erlotinib has been discontinued in use for the most part because it has some properties that people are not very happy with. But I would argue that we should still consider combination treatments with erlotinib, even though it's not typically used now. It may not work as well as osimertinib as a single agent, but this is an incredibly strong selection happening. We get resistance, but if we could do a combination with this really strong selection, that implies that we were hitting a bottleneck and we may be able to actually, the idea ultimately is to drive the cancer population extinct. And this is evidence that a combination with erlotinib might do that. All right, here are the lessons I just told you. Thank you for listening to me talk about that. I want to thank funders over many years that have helped me, NIH, Gilead Sciences, the Natsu Orm Sands Foundation, the J.M.R. Barker Foundation. And the last thing I'll say is that I told you that I was only going to talk about point mutations. We are now beginning to be able to do this for copy number changes as well. This is much harder, but we finally have done it. After 2 years of extremely high level of funding from J.M.R. Barker Foundation to do this, this is just an example of chromosome, and you can see there's all these peaks, and it's like, well, which genes are responsible here? I don't know. And I won't explain exactly what this means, but when we pick particular genes and say, okay, we nominate this one as being selected, we can see that we can basically explain all of the copy number changes that are occurring by CCR7 being selected and by SMARCE1, by SMARCE1, which is a tumor suppressor, being selected not for being amplified. And that's like the whole chromosome can be explained by just those two things. Everything else is just neutral mutations of copy number changes. So we're really at a point where I think we can understand copy number changes and how they're causing cancer in a much better way than ever before. And I'm really excited to combine these two pieces of information and perhaps unify it to explain the causation of cancer at a somatic genetic level. Thank you for your attention.
Thanks, Jeff. Very nice talk. So any question? Question? Okay, I have, yeah.
Sorry.
Go ahead.
It's a fascinating talk. Roberto Pugliama, I'm an oncologist. So do you think this algorithm, you alluded to in the last few slides about the implication cancer therapeutics, but are you advocating to apply this platform as a predictor to response? In disease where we don't have clear oncogenic driver like EGFR or ALK, how we can really predict response?
The answer is yes, but it's complicated. And the yes is these are the causative changes that are leading to cancer. So it should be helpful for outcomes, right? The answer is yes. But I don't believe I don't believe I've exhausted everything. Even if I can get copy number changes in there, there's also epigenetic changes. Perhaps there are transcriptionally stable states. There's a lot out there, right? And the problem is that we're looking at a subset of what's causing cancer. And I strongly believe these major somatic mutation drivers are huge in terms of the causation. Like, I don't want to understate that. But because there's other things out there that are also causative that we don't really have a handle on the same quantitative way, if we take out a tumor at a certain stage, what we kind of already know is that it's had enough of these drivers happen to be a tumor at that stage. So the problem is, if some subset of that is there, that doesn't tell you whether or not you should expect an outcome, because there's a lot of other stuff there as well. So I do believe it has a role, but it's a nuanced and complicated thing. We can't just say, oh, KRAS is a strong driver. It's going to cause aggressiveness and make someone more likely to have a worse outcome. It just doesn't correlate like that. And I think that's for this very reason, that yeah, KRAS can cause almost all of the cancer. That doesn't mean it's going to be more aggressive, because we still pick out the cancer at the time when we notice it causing a problem. And so it's always got that level of problematicity already. So really, it's more about the biology of them. Like, is it aggressive? Is it causing this metastatic change? So the bottom line is, if we choose how we do the analysis of data right, I think it may have more insight into that. So for instance, looking at the stage specific of what causes metastasis, there I think we have some insight, because we're dividing it by the property that really relates to the health of the individual. The growth of the cancer does to some extent, but some people have cancers that grow for a long time. As long as they don't metastasize, it cannot be that deadly. I mean, you still don't want it, but what really, ends people's life is almost always metastasis.
So thank you for your fantastic talk. So I have a question that, so now we have a lot of single cell DNA sequencing. So do you think your framework can apply to that kind of data set to understand the evolution process of mutation more details, or something?
Yeah, it's a really good question. I have to say I have not fully applied my mind to figuring that out. The way that I don't know how well this is going to communicate to everyone, but I'll express it. And if it gives you something, great. If it doesn't, doesn't. My background's in evolutionary biology. So the approach that I sort of have taken that is kind of unique compared to what other people have done in looking at these tumors is a molecular evolutionary approach. In other words, that's an approach where you look at it and you say, certain mutations have happened and then they fixed, and then another mutation happened and fixed, and another mutation and happened and fixed. And now I can look at how frequently those fixed over time, in multiple tumors, and I can learn something about this selection that's happening. What most people in cancer evolution are doing is looking at the population genetic approach, which is looking at, like, here's a time slice and here are the clones and how are they competing against each other. So that's a population genetic approach. And I'm telling you this because your question was, to what extent can single cell tell me the answer? Well, as long as single cell is focusing on this population genetic thing, then the kinds of tools I'm using are not so insightful to it. But in principle, you're looking at the same kind of data just on a more refined scale. So in principle, there should be some way to assemble all of that population genetic data of those single cells and sort of reconstruct this. But what I'd argue is that what you gain out of single cell is mostly a much better piece of data about the population genetics. You don't actually gain a whole lot for what I'm doing, because most of what's driving my result is the things that have already happened and fixed. And so they're fixed in all of those single cells. So I'm not really gaining much from it, which is why I haven't spent my brain power on it as much as one might think, because everyone's gathering single cell data now. I do think, of course, single cell is a great breakthrough, but it's really the so other inference tools that it's going to be particularly strong for, and not for the tools that I'm developing or have been developing.
Thank you so much.
Other questions? I have a couple questions about detecting real gene mutations. So, you see, it's well known that gene mutation has a long tail phenomenon, right? So basically, means a few genes has a high mutation rate, but most of them has a very low mutation rate, right? So then, my question is, how to detect those real, really occurred but functionally important gene mutations?
How do I detect the ones?
Yeah, that's I mean, really occurred mutation, but they are functionally important.
Oh, that rarely occur.
Yeah, really occurred. Yeah.
I mean, I can only use the same data everyone else is, so I don't gain. I have a statistically powerful approach, but when you have very rare mutations, there's very little power to detect them. So I would just answer that by pointing back to the part of my talk where I talked about that prostate cancer distribution and the fact that additional tumor sequence gets us more and more significance, and it's continuing. And even now, when we have sometimes thousands, even some data sets we're working with have 9,000 tumors, some have 15,000 tumors. The more we get, the more data we get, and the more inference we get. And for instance, in those evolutionary landscapes I showed you for lung cancer, where we have lots of data, there's a ton of tumors that have been sequenced, bulk tumor sequenced for lung cancer, we're able to sort of figure out those landscapes. But if you think about it, like as you go down to each division of LRP1B plus KRAS, LRP1B plus TP53, those sample sizes go down very, very quickly. So again, the more of this tumor sequence data we get, the more exactly we can understand these evolutionary trajectories and how these things interact. So I'm just, again, echoing the point that essentially all I think is, as we gather this more data, we need to continue to make it available so that we can analyze it in bigger and bigger data sets, because we learn more and more and more as we do that. And it is not a situation where we've saturated what we can learn from these sorts of sequencing analyses.
Another question is about the evolutionary trajectory analysis. Okay, so if I understand you correctly, basically you perform pairwise comparison, right? Okay, the mutation to the two genes. So whether the occur of one gene will increase the chance of another gene to mutate, right?
I wouldn't characterize it as performing pairwise comparisons, but yes, we do look at pairs.
Yeah, okay.
Yes.
So this is a first order interaction, right?
Yes.
Oh, okay. So I think whether it is possible to do the high order analysis?
Yeah, and that actually is exactly what those evolutionary landscapes were that I showed you, where I had LRP1B and KRAS. That was a higher order landscape, meaning we have the epistatic interactions of every single of LRP1B versus LRP1B plus TP53. So all possible permutations of pairwise and higher order epistatic interactions were included in that map. And so the selection for P53 in the context of KRAS and LRP1B, that's a three entity question. Like, you have LRP1B and KRAS, what's the selection for P53? So yes, we're able to do that. And the thing that stops us is exactly the last answer I gave, which is that as we look at more, it's not the computation. We figured out a very good way. Jorge's a mathematician to actually figure out that information. But the problem is we run out of tumors with higher order interactions as you get to more and more. So we can get maybe 4 or 5 levels of 4 or 5 different genes into one of those diagrams. But if we try to do more genes, it would work in principle, but in practice, we end up with empty cells where we don't have any examples to tell us what things are, what the selection on things are.
Okay, thanks. Any question online? Okay. Okay. Good. So I guess thank you, Dr. Townsend. Okay. Yeah, thank you.